Good evening, everyone. Welcome. I'm Pam Schaff. I'm the director of the HEAL program here at Keck. And I just want to welcome you all to this wonderful event. This is our 7th artist and researcher exhibit. And I think it is our 3rd year, right, Eric? In collaboration with... Third year in collaboration with Roski students, and our 2nd year with Eric's class. And I will turn over remarks to Ted Meyer, our artist in residence, because I want him to share with all of you the reason that we do this. And then we'll hear from Eric as well.
Hi. Too many microphones. So I don't know if I told this story in the class when I met with you guys, but I have a rare illness. And when I was about 5 years old, I gave bone marrow for research, and there wasn't a treatment until I was 42. And I know that a lot of you researchers and all the researchers here work really, really hard in their cubicles, in their labs, and they don't get out enough, and they don't get enough accolades from us for doing the work that they do that saves all our lives, and especially mine. Thank you. So this is my little thank you to the researchers, and let you guys know that you're appreciated. Plus, I love art, and I love the mix of the two, and that's where Eric comes in.
That's where they come in. Well, first I have to take attendance. Let me scan the room. Thank you to the researchers. A big round of applause for the students as well for the work that they put into this. I think I'm a huge advocate of interdisciplinary studies, interdisciplinary approach to research and science. I think it's one of the great advantages we have of being at a major research university is that scientists and designers and artists can come together in such a meaningful way. And the results are, I'm always impressed. I'm always so proud of all of the people who did all the work in my class. They did the work. I just provide some guardrails.
Hello, everyone. My researcher is Dr. Aram Modrek, and he is an assistant professor of radiation oncology here at USC Keck. And his research basically focuses on how radiation therapy can be used to target cancer cells in the brain. So I interpreted his work in the style of a sci-fi movie poster or comic poster. And this was done when I was talking with Dr. Modrek. He kind of mentioned how he felt like his work felt like science fiction. And this really resonated with me. I felt like this would be an interesting way to portray his work, which most people don't see. You know, it's in the back, it's in the research. It's not like interacting with patients. So his research specifically focuses on why some cancer cells are able to survive the radiation therapy. And once they survive the radiation therapy, they're actually more resistant to future treatments with the radiation therapy. So that just sounded like the perfect sci-fi villain, like surviving the thing that's supposed to kill them, making them stronger. So here I depicted the radiation therapy as like a ray gun, sci-fi ray gun. And then zapping what's supposed to be like the evil cancer cells here, but then one of them turning into like the super mutated cancer cell. And I think it's just a fun way for people who might not have like a science background to kind of get a better understanding or a better perspective of what is actually going on through his research. And I think it's a way to depict that it's an actual battle against cancer through using his radiation therapy research. And I want to say that I had a lot of fun making this process. It was really interesting getting to know your research, and I thank you for the entire process and what you're doing. Thank you.
Thank you so much. And I didn't do much here. This was all him. And thank you for all the time and hours and back and forth you went into to understand everything and put in your own creative input. I really love it. Very proud of you. And I was staring at this. I took a bunch of pictures and I'm going to show everybody. Congratulations.
Thank you so much.
Thank you. Thank you, everybody. I think we're missing one artist, but this other piece of work is also quite beautiful. I don't want to speak for the artist, though. Yes, there's two pieces here. I think they went in two different artistic directions. It's kind of funny because they're contrasting too color-wise, and it's they're both beautiful. So yeah, I've been staring at them across the room. Yeah.
Hi, my name's Olivia Cartaya.
I'm Cassidy.
Yeah, I'm Danny Wang.
So as a group, we learned that Dr. Wang's team actually works with black blood MRI imaging in the brain. And essentially that is a completely new technology that us in the regular hospitals and clinics have yet been able to see. And this imaging is so detailed and gets so precise that they're able to get to what, the nearest micrometer or crazy small amount that we can't even fathom with our own brain. And so Cassie and I set out to kind of explore that when we do take these scans of the brains, it's in different sections, and also just like the finite details of the actual blood vessels within the brain, and the actual like importance of that. This imaging is currently being used in studies in vivo, like with Danny's team, but bringing awareness to this type of technology will help bring it into actual patients and actual use, and actually help people either struggling with brain injuries, stroke, all sorts. And this was just a wonderful collaboration between Cassidy, Danny, and I had a great time. And I thank you so much for participating again, Dr. Wang. I know this is multiple years in the making of you being a participant in this, and we're glad to make another piece to add to your collection.
Thank you so much, Olivia and Cassidy. This is a masterpiece, I would say, definitely. I think it's very intuitive. You can see each cross-sectional and also dynamic, I would say. So yeah, I think it should go to Metropolitan, but I'm happy to, into my collection, if you allow me. Thank you so much.
Alrighty. Hi everyone, my name is Christian. I'm a graduate student here at USC Stem Cell. My artist was Yujin. Unfortunately, she was unable to make it today, but I did ask her for a speech of what she would say if she was here, so I'll speak that on her behalf. Before again, I just want to first say, obviously, thank you to Ted, Pam, and Eric, obviously, for creating this. This is my third year doing this, and it's always been inspirational and beautiful to see the work people have done. Unfortunately, it's also my last year as I am departing from the university, so I will be sad to see this go. But once again, I think great work for everyone here today. So in terms of my science work, I work a lot with computational biology and developmental biology. Essentially, the premise here is, can we create simulations to predict and actually model biological and tissue functions that exist in reality? But obviously, I don't want to speak too much into the science. I actually wanted to read off of what Yujin had to say. So I think it's pretty beautiful in itself. So from her, she said, this work serves to explore Chung's research methodologies. In his pursuit of studying synthetic morphogenesis, he has found a unique intersection of innovation and biology. Using an interface language that is familiar to many of us, I mimic the nature of a desktop screen, with folders scattered across. The main working component of this piece are the real flowers inside the folders, which were intended to bring out the contrast between the digital and physical element of the research work. The living elements parallel the in vitro methods of testing with living organisms. The digital components parallel the in silico methods of using computational biology. I wanted to highlight the work of both working physically in the lab and also working with computational biology, because I found it commendable that Chung had developed an entire coding system specifically meant to test and simulate the functions of the cells he is studying. The wallpaper background of the desktop is composed of binary code and ASCII symbols to represent the computational nature of the work. And these characters create an image of the cells obtained in his lab, which was obtained through a microscope. And fun fact, it's a little bit nostalgic for me because the picture you see in the background was actually the very first cells I've ever imaged in my life back in like 2020 when I was 18 or something. So kind of nostalgic for me. And then she finished it off by saying, together, I use these components to develop the representation of computational biology and to reflect its reality in the real world. But again, so unfortunately couldn't make it today, but I just really want to commend her for the work. I thought it was a beautiful piece. And yeah, I'm happy to hear from everyone else here today as well too.
Hey everyone. So this is our team. I'm Yongtae, and that's Isabella, Melanie. So our work, our researchers' work focuses on synthesizing tissue in a user-defined way using programmed stem cells. So what that means is by encoding ways to interact with other cells, we can make tissues in a specific form we want. And from our discussion with Dr. Leonardo Morse, we saw the huge potential of his research in advancing the regenerative medicine field. And during our conversation, he also mentioned that he wanted to express his work to his child in an understandable way. And from that, we thought it would be very cool to make work that would inspire the future generation by explaining his really cool research in an easily understandable way through visual metaphors and cute-looking characters that clearly explain his concept in a really easy way. So this was a 3-person project. I took the lead in the scripting, initial sketches, and the final layout. But I want to give credit to the amazing artwork that Isabella and Melanie did. If you'd like to talk about your artistic process, that would be really nice.
Oh, yeah. I was responsible for the characters and all the little, everything that's basically in the foreground here. The goal was just to make them cute and appealing and make the visual metaphor pretty clean and easy to understand immediately. The thought was, you know, who says that pretty complex biology is not for children? You know, I feel like to make it accessible to people that don't have a science background, it's pretty important to make it welcoming first and easy to read.
Hi, I'm Melanie. I worked on the backgrounding, and my goal overall was just to create a setting that was inviting and like able to be readable for the children potentially reading this comic strip. Yeah.
Hi, my name is Jiwon, and my other partner was Chloe, who is unfortunately not here due to classes. But for my researcher, it was Dr. DJ Cast, and she is the JEP's Director of STEM Education. So she's done a lot of efforts in providing accessible STEM education to underrepresented communities out in the LA community. And we kind of wanted to encapsulate what would be interesting towards elementary school students, and that was kind of towards the comic book poster selection. So when you go to the book fair as a kid, you go to see all the different posters and you're really attracted to them. So we kind of wanted to encapsulate that sense of nostalgia and wanting as a kid, but reflected in STEM careers. So we wanted to represent different careers in STEM, such as like an astronaut, an engineer, or a lab scientist, and allow the students to see themselves in these posters, because representation is a very crucial thing as a kid, and being able to see yourselves in these posters can also motivate you and inspire you to pursue those careers as well.
This was my first time working with this particular project, and I think it was a really fun way to bring all of these scientists and these artists together. As a STEM educator, I felt a little out of place, but I really appreciated the view that Jiwoo took in terms of, like, you know, the students are our future, and, you know, that's why it's italicized. And I love that you also use some of the faces of the students in our program in this work. And I can't wait to like mass print them to get them actually into some of our classrooms as well.
Hello, my name is Ketcher Salazar. I'm an undergrad in Dr. Morsuit's lab, and we research synthetic biology, and essentially we try to reprogram cells to do stuff that they normally wouldn't do. And the twist with my research and Dr. Morsuit's research is that we're doing this in mammalian cells while it's usually done in bacterial cells. And the point of this is that we could actually try and build tissues and new structures that don't usually occur in nature to regenerate tissue and specifically synthetically elongate structures, which is what I specifically research. So if you can imagine, hopefully far down the road, if you cut off your finger, cut off your limb, the research I do now could help you remediate that.
Hi everyone, I'm Zoe. Layla and I are the two artists that were working with Catcher to bring his research to life in a more artistic way. And to us, the philosophical foundation of Catcher's work could really be boiled down to two things. One, bringing things to life that doesn't yet exist, and seeing life in a completely new light. And those were the guiding principles that we thought of when trying to make this multidimensional art installation that Layla can talk more about.
Yeah, so most of our process was revolving around the fact that we wanted to visually embody Catcher's work. So thinking about us going back to the studio, a lot of his work involves looking at cells and using digital imaging, fluorescence digital imaging to depict pictures and see colors to actually see if the cells are elongating to create those structures. So keeping in mind the color, keeping in mind the fact that this is something that isn't occurring in nature yet, it doesn't really exist yet, and just the imaginative aura that exists within Catcher's research, we created this piece. We used a ton of color, acrylic, and we just wanted to basically show how the light reflecting off of our piece can create something new. And that's what we get from the shadow in the piece. But that was our mission with the artwork.
Hi everyone, I'm Allison and I'm working with the researcher Christopher Shira. And essentially he does research on the ear. It gets very complex very fast. So when talking to him and asking him like what is like the biggest thing that you want people to learn and understand from the end of this piece, it was the idea that your ear can also produce sound. So a lot of people know that the ear like receives sound and it's how you hear, but not a lot of people know that it also has its own unique sound. So everyone here, for the most part, hopefully, is able to produce sound from their ear. So I decided to create like essentially like a blueprint of the ear, pretty much going into detail and showing like the cochlea in a 3D rendition. Because a lot of times when I was doing the research and I would look for like images of ears, it's not the most beautiful imaging of it, and they're actually like blueprint designs in general are never used for like showing the body or just anatomy in general. So I thought this would be a unique direction to show something that's pretty scientific and detailed. But yeah.
Yeah, thank you, Allison. I think you did a fantastic job here. I love the idea of the blueprint. And I think it captures what we do in the lab really well, because what we do is we listen closely to the ear. And these sounds that the ear makes are basically the byproduct of little so-called hair cells in the ear, very different from the hair cells that are depicted over here. And these hair cells are piezoelectric in their action. In other words, they move when an electric field is applied across them. And as a result of that, the ear also makes sound while it's listening to sound. And we use those sounds to try to figure out how these piezoelectric elements work, and also to try to figure out how we can better diagnose different types of hearing impairment. So congratulations, it's really beautiful, thank you.
Hi, my name is Sheesh, and I don't think Dr. Chen is here yet, but basically she was introducing me like 13 different research topics, but we decided on the per and polyfluorinated substances that have impacts on our health. And basically her research topic kind of focuses on how this chemical substance has specifically on women and unborn child. And dubbed Forever Chemicals, it's a growing concern for epidemiologists and at the public large, because like all the health problems that we get impact from, like just drinking water, and like and how it's not very persistent in the environment, and it's not naturally biodegradable. So it's with us every single day. We
consume it every single day, and in the future it'll become a bigger problem. And basically, Dr. Chen is trying to help on how to improve and degrade that process, chemical process. And through this AR work, like you can't see it right now, but basically I touched up on the chemical structures and the cycle from source to exposure of the chemicals, and a visual graphic of like how and what the unborn child can have on their developmental process. Yeah.
Okay, I'm Julia Schwartz. I'm responsible for the collaboration with both Matthias and Mina. My lab work on skin stem cells, and some of the things that we are trying to understand is how skin stems are being regulated to help us heal wounds and grow our hair. And some other thing that we talked about is a new interest in our lab is what happened to these stem cells as we age, and some differences between men and women, and why we are now exploring some of the reason why men and women, their skin age differently. And I'll give the stage to the actual people who actually did the work.
Thank you, Julia. So for my work, Julia, me and Mina went to her office and interviewed her for about like an hour and a half, 2 hours, and she gave us a beautiful explanation of all her research and everything to do with it, as she just mentioned, with aging. Beautiful explanation. It just happened to take like an hour and a half long, which is great. But one of the things that we were trying to accomplish with my piece in particular was to communicate her research in an effective and quick manner. And I decided to go more with the hopeful future that her research will bring into the world, not having men bald at the age of 65 or 70, and hopefully continue to have luscious locks. So it just starts off. Another thing that we clearly identified that we had a strong connection with was the idea of narrative and storytelling. And so that's something that we wanted to push all the way across. So you'll just follow me. It's a guy with no hair, and then it's a little stem cell watering the plant that is the hair, and then the hair grows, and now he has beautiful long hair. So it's just a quick little explanation that gets people involved in the research and shows the hopeful future of it. Pass it off to.
And my part of the work kind of focuses on how Dr. Schwartz's study on skin's healing properties could be used as more data visualization points, and how can it be interpreted in that way. So my first part of my project relies on how she utilizes her sensory neurons and skin cells interact in a way that synapses and messenger and receivers interact to create more healing properties. And the second part of it focuses on how chromosomal and hormonal factors affect healing properties, and how men and women have different factors and during these healing. So the first part shows that when estrogen levels are higher, there's a higher skin healing. So I kind of wanted to create a data visualization point on her research, longitudinal research. So yeah.
Hi everyone, I'm Manasa.
I'm Diego.
Do you want to explain a little bit about your research before we get into this?
Yeah, sure, I'll do it. So I'm Diego. I'm a fourth year PhD student in Dr. Offering's lab, where we study lung cancer and trying to develop therapies for lung cancer. And so our project is inspired by a response in patients, about 15% of patients. And we look at that response and we ask how those patients survive longer, and how can we extend that to all the patients, so all of these patients could have extended and improved survival. And so that's what my project is, and what Manasa did the art based on.
Yeah, so Diego was kind of to give me a very thorough explanation so I could understand properly what was happening with the research and the DNA and the proteins especially that they're working in this process to find an alternate treatment to small cell lung cancer. So using that finding, which I felt was at the core and the foundation of the research that I believe happened in the 80s, That's what you said. So using that 15% who are exhibiting this autoimmune response, I essentially want to turn that into a sort of data visualization art installation piece. So you'll find here the 100 Petri dishes to represent those 100 small cell lung cancer patients. And in 15 of them, you'll find these fluorescent pieces, essentially to represent that immune response, which is at the core of the research and what they're trying to do.
Hi, my name is Dr. Martin Kast, and I've been paired with Sam Farrow for this exhibition. I study human papillomavirus and especially the link to cervical cancer. And in 1951, a remarkable event took place. A woman entered the hospital of Johns Hopkins in Baltimore, and she was diagnosed with cervical cancer, operated, the cancer was removed, and the surgeon thought, why don't I try to see if I can culture her cells in the laboratory? And he was able to do it. It became the first human cell line ever that was able to grow in the laboratory. And her cells are still growing. As long as you feed them, they keep on proliferating, and I could fill the whole building with her cells. So that means that in a way, she is still alive. And she is therefore immortal. She's also nicknamed as the mother of modern medicine because her cells have been so important for medical research and still are. They're being used in thousands of laboratories. The interesting thing is that her children thought that she was still alive because her cells are still growing. And then where is your spirit? Is it in your brain? Is it in your heart? Is it in your genes? Her cells are still growing. So in a way, she is truly immortal. There is a book called The Immortal Life of Henrietta Lacks, written by Rebecca Skloot. There is a movie, The Immortal Life of Henrietta Lacks. Oprah Winfrey plays the daughter of Henrietta Lacks. Very recommended to read both the book and see the movie. And so when I was paired with Sam, I talked about this particular woman and how important she was, and that she, in a way, was still alive. And you have done a remarkable job by seeing this one painting here, or sort of painting, but you can see it in two ways. And you will now explain what you actually see.
Yes, so I wanted to highlight Henrietta Lacks, as we talked about. So if you hold up, you'll see the two lenses, and you'll see two basically different versions of the piece. You'll see her portrait, and then underneath that, you'll see the skeleton, the flower crown, and the heart. And I really wanted to honor her spirit and represent that. So each flower, depending on the culture, represents immortality. So just there's little pieces in it that, really highlight her life, the history of it that came with it, and what she's done for modern medicine. Yeah.
Very good.
Hi, so my name is Caroline, and my partner was Lana, but unfortunately she's not here tonight. But I'll do my best on behalf of both of us. And our researcher was Dr. Ideo Fringa, who is also unfortunately not here tonight with us. But
I'm Sophia. I'm a postbac at Dr. Fringa's lab, so I'm here on behalf of Dr. Fringa.
So when we were first meeting, me, Lana, and Dr. Fringa, we were all talking about Dr. Fringa's work. She studies lung cancer, specifically small cell lung cancer. But as we were talking, she got really passionate talking about how there's really a huge lack of awareness and funding surrounding lung cancer. Lung cancer actually kills more people annually than other forms of cancer, such as prostate, breast cancer combined. Yet it doesn't have nearly the same amount of funding or awareness that those forms of cancer do. So as we were researching her work, we actually found out that she makes a lot of TikToks surrounding lung cancer, and I was like, you know what, I love that. As we were talking, it became really obvious that she's passionate about really making this serious topic that seems very complex, very digestible for young people, and that's why she was making these TikToks. And so when we were talking, we decided that we wanted to create a series of posters or infographics that brought awareness to particularly young people, especially surrounding smoking and vaping. So Lana and I each chose a topic. Hers is surrounding screening. Actually, what we found out was that
California ranks dead last in terms of how many people get screened for lung cancer, and we've been ranking consistently last. So that was definitely something that we wanted to touch on. And then mine just overall goes over general facts surrounding lung cancer. But one thing that we really wanted to do was make it accessible and visually, draw in visually people that are young. And so we both brought our unique art styles to it, and that's what made it really fun. We were able to create these infographics, but still bring our unique art styles to create something that draws in young people visually.
So my work is a 5 illustration box that the users, the audience can take down and read the back of it. So this piece is about how kids wait in the waiting rooms just see hospital. So my researcher called it chaotic symphony because every doctor knows what they want to do, but it's still very chaotic. It takes no time. So this piece makes kids want to interact with each piece and knowing what each step they're doing.
I just want to say, we've done this 7 times now, and last year was stellar, and this class does a great job, which is a testament to you guys. This looks really professional. The variety of materials, it says a lot about you. It says a lot about your teacher, that it looks so great.
I just want to second that and say I am so impressed, especially as a storyteller and a student of literature, the stories that have been told here and the ways in which your interpretations have improved the way that people can understand the science and can really relate to things that may not otherwise be talked about or understood so well. And I can see in the room things that I believe are going to go out in the world and make a difference. So I'm really thrilled to have experienced this, and thank you, Eric, to the guidance you provided to your students. Thank you, Ted, for inspiring us to do this work. And I hope everyone will continue to make your way around, study the pieces, please eat some food, and enjoy the evening. And thank you.